Good morning, everybody. It's a pleasure for me to be here today. I'd like to start by thanking the session chairs for giving me this opportunity to speak to you today about the work that we've been doing in the lab, looking at single molecule imaging of cytoplasmic dynein inside living cells to reveal the mechanism of motor activation and cargo capture. So as most of you in the audience are already aware, cytoplasmic dynein, like most of the motor proteins, is regulated for its activity. And cytoplasmic dynein, in fact, requires the formation of a tripartite complex for its processive movement inside cells. A lot of work has been done on this in vitro. So if you look at recombinant dynein on microtubules in vitro, you see that dynein doesn't really walk processively on its own. But once you add dynein's regulator DYNACTIN and a cargo adapter such as big D2, you start seeing that dynein behaves as a very nice processive motor that moves towards the minus ends of microtubules. So the question that we set out to ask was, how does this tripartite complex actually come together within the complex intracellular environment to actually activate cytoplasmic dynein and to initiate cargo transport? So to answer this question, Nirikshit, who was a graduate student in the lab, went ahead and optimized highly inclined laminated optical sheet microscopy for visualizing single molecules of dynein in living HeLa cells. So these are dynein heavy chains tagged with GFP. And what you'll see is that there are these molecules that are immediately visible within the cytoplasm of these living cells. And while you see that there are diffusing molecules in the cytoplasm, there are also instances of molecules that transiently stay in the field of view for the duration of the movie, for a short duration during the movie. And what we first did was to go ahead and confirm that what we were looking at were indeed single molecules, and that they were binding to microtubules when they actually remain in the field of view. So we can track these molecules that appear afresh on the microtubule. And if you take a look at another such movie, what you see here is an instance of dynein. In the white arrowheads are pointing to an instance where dynein is binding to the microtubule and not moving very much upon its binding. In contrast, the teal arrowheads here and green in the movie are indicating an instance of dynein binding to the microtubule and moving towards the minus ends of microtubules, which is towards the nucleus in this instance. And conversely, we also have molecules which bind to the microtubule and move towards the plus ends of microtubules. And these plus end events we think are likely that of dynein binding to cargo that is already being moved towards the plus ends by a kinesin. But the majority of events that we see are of this kind, which are stationary. More than half of the events are actually of stationary dyneins on the microtubule. And a small population, a third of the molecules, actually move towards the minus ends upon binding to the microtubule. And this is actually confirming what others have seen in vitro settings, which is that dynein on its own typically is in a default inactive state. We presume this is what is happening in this instance as well. So what we then went on to look at was the duration of time that dynein actually stayed on the microtubule. Interestingly, what we saw was that, what is also apparent here, is that dynein does not necessarily undertake long-range movement upon its binding to the microtubule. Rather, what happens is that it typically stays on the microtubule for a very short duration of time, only of the order of half a second, a little over half a second in fact, before it detaches and moves back into the cytoplasm. And this is an interesting suggestion because in vitro, you see the dynein, when it does move processively, it does actually move quite a long distance before it attaches. So I'll come back to this later in the talk, but for now, what we conclude by looking at just dynein on its own is that dynein interacts transiently with the microtubules, and it detaches and attaches to the microtubule frequently. The second part of the tripartite complex is DYNACTIN itself. The DYNACTIN is a multi-subunit complex, which includes the P150 subunit, which has an independent microtubule binding domain. So we looked at P150 in our cells. So we looked at this using surf microscopy. And what we see is that DYNACTIN's localization, P150's localization in this case, is along the entire length of the microtubule. So there is this theory in the field, which is that DYNACTIN accumulates at the growing plus tips of microtubules because of its association with the EB1 and CLIP170 complex. And because of DYNACTIN's clustering at the growing plus ends, dynein also clusters the growing plus ends of microtubules and therefore is able to capture cargo at the cell periphery. But what we saw here instead was that rather than only accumulating at the plus tips, in fact, only about 16% of microtubule plus ends had an accumulation of P150. We saw P150 predominantly along the entire length of the microtubule lattice, as is quantified here. So too, we wanted to make sure that the P150 we saw was part of the entire complex and not just P150 on its own, because like I mentioned previously, P150 on its own is able to bind to the microtubule. So to do this, we looked at another component of DYNACTIN, which is P62. So we saw that P62 occurs very frequently with P150 on the microtubule. And therefore, we concluded that there is likely a very high possibility that the entire DYNACTIN complex is present along the entire length of the microtubule. And what I have not shown here today, but we went ahead and confirmed this in live cell imaging experiments, is that DYNACTIN actually remains on the microtubule for a much longer period of time than dynein does, for the order of minutes for that matter. So essentially what we saw is that DYNACTIN, which is the second component of the tripartite complex, is persistently bound along the length of the microtubule. So the last part of the tripartite complex, of course, is the cargo adapter. Here we use the cargo as a proxy for the cargo adapter. And we looked at two different kinds of cargo, which undertake net minus indirected movement. So one is dextrin and the other is EGF. So both of these are fluorescently tagged and the cells are allowed to take up these cargo, and we follow the movement of these cargo thereafter. So both dextrin and EGF actually end up at lysosomes at these concentrations that we've used at least. And the interesting thing is, from previous studies, it's known that dextrin takes about the order of hours to reach the lysosome, whereas EGF takes the order of tens of minutes. And what we first did was to visualize the movement of cargo over a period of about 2.5 minutes or so. And what you can see immediately here is that the movements are quite different for both dextrin and EGF. Whereas dextrin has seemingly a lot more pause state movement, pause states, EGF seems to be moving a lot more, as is evident from the chemographs. We were able to quantify this difference and saw that, in fact, the net, the uninterrupted minus n runs for each of these cargoes is actually quite similar and only of the order of a little over half a second. On the other hand, if you look at the mean pause times for dextrin versus EGF containing endosomes, the pause time of dextrin containing endosomes was a third higher than that of EGF, which of course makes sense given the chemographs that we see here. And another thing I'd like to point out here is that the minus n run times of both dextrin and EGF actually match very well with the residence times of dynein on the microtubule, which we saw just a few slides ago, which is about 0.7 seconds. So essentially what we see here is that these two different kinds of cargo have very different kinds of trajectories and pause times. And of course, the person that did all of this work was Greg, who's a postdoc here at UNSW. The second thing we asked with respect to the cargo was where it was in relation to the microtubules as well as the cargo, as well as DYNACTIN, sorry. And what we see here first is that the cargo remains close to microtubules regardless of what time you look at it. So we visualize these in live cell surf microscopy experiments as well as in correlated light and electron microscopy images. And this is work by Natasha and Nick at the EMU division here in UNSW. And also with respect to DYNACTIN, the cargo actually remains associated with, here we're looking at P62. So all of the early endosomes we looked at here were, majority of the early endosomes we looked at here were bound to P62. So what we see here is essentially that the cargo is positioned close to microtubules and DYNACTIN at any given point in time. So the question we asked next was, putting all of these together, can we have a picture emerging wherein we see that dynein is able to attach to cargo and DYNACTIN positioned close to the microtubules and enable a short minus n directed run on the microtubule? And for doing this, we actually had to do a bit of hard work with respect to our microscopy, because this is quite a challenging experiment. But essentially we did 2-color imaging, high-low imaging, wherein we're looking at both the cargo as well as single molecules of dynein. And when we do this, we see instances where the cargo, which is initially stationary, is bound by a single molecule of dynein and moved towards the minus end in a very short run. And this is true with dextrin cargo, but also with EGF containing endosomes, as you can see here. You'll see that in the movie as well with the arrowhead. And it's quantified here in these plots with the intensities of the dynein molecule in green and the position of the cargo indicated in magenta. As you can see, there is a movement only after dynein binding, and as soon as dynein detaches, you see that the cargo remains stationary. So this is interesting. Of course, the question was, can this kind of movement actually lead to long-term movement inside the cell for cargo that are required to move over long distances? And this is where we went ahead and looked at EGF as a cargo. And this is a movie over several tens of minutes. And what you see here is the actual entry of the cargo and subsequent movement towards the minus ends of microtubules. So if you looked at these movies and sampled at every 5 seconds, you'll see movement which is very familiar to most of us in the field that look at endosome trafficking. You'll see that there seem to be nice long-range directed movements inside the cell. But if you actually now dial down and look into sampling at every 0.5 seconds, you start seeing these kinds of movements that are very reminiscent of what we've quantified earlier, which are these intermittent runs interspersed with pauses. To make sense of all of this, we actually teamed up with the physicists Vijay Kumar Krishnamurthy and Prita Dolai at the International Center for Theoretical Science in Bangalore. And what we did was to actually build a stochastic motor cargo dynamics model, or an MCD model, which incorporates individual motor dynamics that we measured in our system. And what we were able to do was very accurately simulate trajectories of cargo for both dextrin and EGF endosomes. And we used these trajectories of cargo in a coarse-grained 3-state run-and-tumble particle model, which is accurately able to predict emergent cargo behavior, as is shown in these plots. So essentially what we've shown here is that with data that we've used taken from our experiments, we've used them in these MCD stochastic model. And by just changing the on rates of dynein to the cargo, there is a third higher on rate of dynein to EGF containing endosomes than there is to dextrin in our model. And this is able to accurately then predict what the cargo behavior is like in our system. And this is actually quite powerful because we could potentially use just the cargo trajectories to infer the behavior of individual motors in our system. This is essentially the model that we have. The updated results are actually online on bioRxiv. Please do check out our paper. And I'd like to end by thanking all the people that have been involved in this work. This is our funding. Thank you, and I'm happy to take your questions now.